Hi there, this is Mr. Wartsky, and today we are going to start our fungus screencast. I'm a fun guy. Are you a fun gal? Um, Make sure if you have any questions, you can pause the slides to make sure that you get all the information down. If you have any questions, please refer to your textbook, or you can watch the video again, or you can ask me in class tomorrow. So we're going to introduce fungus as heterotrophic sap probes, which means that they live on top of the organisms that their food source, live on top of their food source. They produce enzymes which are excreted from the cells. They break down the material outside of their bodies, and then they absorb the nutrients into their cells. Now, fungus are defined by their type of cell wall. They have a chitin cell wall, which cannot be digested by us, as beta glycosidic bonds. They have a suffix as mycota or mycetes, and that's from the Greek myketos, meaning fungus. They are decomposers. They also produce bread because they produce CO2 through anaerobic respiration, fermentation, alcoholic fermentation, by the way. And the alcohol is burned off in the baking process. They produce alcohol, obviously, antibiotics, drugs like cyclosporin, which suppress your immune system so you can prevent the body from rejecting certain things. They produce foods like blue cheese. They also produce diseases like Dutch elm disease, athlete's foot, and ringworm, which ironically is not caused by a worm but by a fungus. Their body of the body of the fungus is called the mycelium, and that's the feeding body, and it's made up of thread-like structures called hyphae or hyphae. They are glued together. Now there are two types of hyphae. There are the ones that have cell walls between the cells, and those are the septate hyphae, and I'm pointing to an example right down here. And the choanocytic hyphae, they have no cell walls between the cells, so they have a continuous cytoplasm, and there's a picture down here of a choanocytic hyphae. It'd be good to draw the picture. It's always nice to refer to it. Now, the hyphae can specialize. They have sporangiophores. That's a type of hyphae that produce sporangia. And they're the sporangiophores right down here, produce the sporangia. And the sporangia produce spores. They have haustoria, which grow into living tissue. So if this was on top of your skin, like the athlete's foot, this would be the haustoria, or the rhizoids, which are hyphae that grow into other food surfaces, supposedly that are dead, like bread or decomposing material. So those are hyphae, and those all get together to form a mycelium, which is the feeding body of the fungus. So fungus, they reproduce sexually, and they reproduce by the fusion of the gametes. Now, fungus have an interesting thing. They separate the fusion of the plasma or the cytoplasm, which is called plasmogamy, and then they separate that from the fusion of the nuclei, the haploid nuclei, which is called karyogamy. So in fungus, you will have the cells fuse, you'll have the gametes fuse, and they'll have plasmogamy, but they will delay karyogamy. So basically, they will have two haploid nuclei in the cell. Now, it's called a dikaryon because it's N plus N. It's technically not a diploid because the two haploid nuclei have yet to fuse. They behave as a diploid, but they're not really diploid, so they're dikaryon. Eventually, the two haploids will fuse and they will undergo immediate meiosis to form four haploid nuclei. And here's a picture of these two cells fusing, and this is plasmogamy. But you'll notice that the two haploid nuclei here will not fuse, and that so they have delayed karyogamy. Now, eventually, when they do fuse, they will go through immediate meiosis. There are 5 divisions of fungi: Chytridiomycota, zygomycetes, which you know as bread mold. Ascomycetes are sac fungus, aspergillus, and yeast. You might remember aspergillus from the AP lab. And yeast, you know about yeast. Basidiomycota are club fungus. No, it's not a place where fungus go to celebrate. They're also known as toadstools and mushrooms, and they're shelf or bracket fungus that grow on the side of trees. And Deuteromycota, which I love the name, fungi imperfecti. We'll talk about those, but they have sexual reproduction. We just haven't seen where it happens yet. Now, all these divisions of fungi are where they have sex or where sex happens. So zygomycetes, their sex happens in a zygospore. Ascomycetes, their sex happens in an ascus. Basidiomycetes, their sex happens in a basidia. And Deuteromycota, we don't know where it happens, but we know it happens. So Chytridiomycota is a link between fungus and protists. They have unicellular, uniflagellated sperm, which is not like the rest of the fungus, but they do have a chitin cell wall like fungus. And that's why they're defined, or that's why they're placed in fungus, because of their cell wall, and they have coenocytic hyphae. And that's pretty much all we're going to say about Chytridiomycota, because I don't know that much. If you go to college and study fungus, by all means, come back and teach me. Zygomycetes bred mold. They have sexual reproduction. So we're going to start off with a haploid spore landing and germinating on a food source. The hyphae grows in all directions. They form rhizoids, which remember are the hyphae that grow into the food source. They secrete enzymes. They break down the food. They absorb it, and they also anchor the mycelium or the feeding bodies. Right? And then they produce sporangiophores, which you know are hyphae that produce sporangia, and the sporangia will produce spores. Hopefully you got that. Now, each zygomycete is a mating type plus or minus. We really can't call them male and female. They're plus and minus, and the opposites attract. So the rhizoids will produce a hormone which will go out and will attract the opposite mating type. So the rhizoids of opposite mating types of pluses and minuses will start growing towards each other. That's spread mold. Now, the opposite rhizoids, their hyphae, the rhizoids will fuse, which is plasmogamy, and they will form a gametangium, and that's like a little starting to form a little ball, and then each cell has two haploid nuclei, and the cells will divide and divide and divide to form a zygospore. But each cell in the zygospore will have two haploid nuclei. Eventually, the nuclei of all those cells fuse and they go through immediate meiosis, but all the spores except for one will die. So only one haploid spore, resulting in the sexual reproduction, survives. And then that spore will go through mitosis and cytokinesis and form a hyphae. And that is the sexual reproduction of a zygomycete. So the sex happens in a zygospore, which I'll show you in a second. The asexual reproduction, the zygosporangia, or the zygosporangia, will produce haploid spores by mitosis, and those spores are released into the environment, and then they will land on food. So we have the life cycle here. So we have, this is the asexual right here, the spores of sporangium, and they will land and grow into hyphae by mitosis. Now here, the rhizoids, right? Rhizoids grow, and the rhizoids grow, and they fuse, and they form a gametangia, right here, right? And the cells here are dikaryon in that they are N plus N, and then eventually the nuclei fuse, and all of the spores except for one die, and then they will grow by mitosis and cytokinesis and form their very own zygomycete. That right there is a zygospore, or that's a sporangium produced by sporangiophore, sporangium. I don't know if you can see little dots. Those are these spores. And they're haploid spores produced by asexual reproduction. So we have the two rhizoids touch. They form a gametangia here, and then the cells divide to form a zygospore right here. Now each cell in the zygospore is dikaryon. Eventually, the nuclei in each cell will fuse. They undergo meiosis. They form haploid spores, and all spores but one die. How do you know which one lives? I have no idea. The third division are ascomycetes. These are sac fungus. They have sex in the ascus, or I like to say sex in the sac. They include morchella, which is edible, blue-green molds, not edible, yeast, aspergillus, and ergot. We'll talk about ergot in a second. There are 60,000 species and 25,000 species of lichens, and we'll talk about lichens later. Ergot can produce drugs that cause hallucinations. Ergot can grow on rye bread, and wet years have been tied to an increased witch accusations in medieval Europe. Ergot can produce lysergic acid diethylamide, and that is LSD. And here are some pictures of ascomycetes, in case you didn't know what they look like. They look like they have a little cup there. So ascomycetes, again, it's going to sound familiar to zygomycetes, their life cycle. Their haploid spores land and germinate and produce hyphae. They grow by mitosis and cytokinesis. In asexual reproduction, some hyphae will produce kinetiophores, which produce kinetiospores, and they produce them by mitosis. Basically, they produce spores. The ends of the hyphae form spores. They don't have a sporangium like the zygomycetes do. Same thing with their sexual reproduction. They have plus and minus hyphae that are attracted to each other because of hormones. Their cells fuse. That's plasmogamy, and their cells will undergo reproduction, asexual reproduction, to produce hyphae that are dikaryon. The hyphae will get together to form an ascocarp, which is the mycelium. And at the cells at the end of each hyphae, their haploid nuclei will fuse, undergo immediate meiosis, which is sex, and form 4 haploid nuclei. Each will form a haploid spore, which will go out into the environment and land and germinate on their own. So we have a diagram of this here. We can start anywhere. It's a cycle, but we'll have the spores. They grow into hyphae. Now this is the asexual. They produce kinetiospores at the end. Or they can grow into long hyphae, which can fuse to form this right here. This is your mycelium. And at the end of the mycelium, at the end of these hyphae here, they undergo karyogamy. They produce now a diploid cell, which undergoes immediate meiosis to form 8, and well, the meiosis would, and they form mitosis to form 8 ascospores through sex. Remember to pause it if you need time to draw it. Now, yeast is an ascomycete, it's single cell, and asexual divides by budding, which means that budding is cytokinesis with unequal cell division. So you have a small cell growing off of a larger cell. It's like if a little person were to grow off the side of you and then fall on the ground and then grow into a big you. Now, the sexual, you have haploid cells that fuse of different mating types. They fuse, their nuclei will fuse. They undergo immediate meiosis. They form 4 haploid nuclei, which can form a haploid cell. The interesting thing about yeast is that they can survive both in with a haploid nuclei or a diploid nuclei. Now, the only ones that can undergo sex are the diploid ones. So you have to have the fusion of 2 haploids to form that diploid in order for them to have sex. Remember to pause if you need to. So why sex? So I always like to say that when the going gets tough, the tough have sex. So sex provides variation in the population. The more variation you have, the more likely some of those organisms will survive harsh conditions. Fungi are defined where they have sex. We talked about that. And we're going to talk about basidiomycetes next. So I hope this helped. Now, if you have any questions, please watch the video over again, read your textbook, or ask me tomorrow in class. Have a good night.